Yeah, pilots. So it looks like that move just ended. So
Okay.
Zero should start to swing in maybe a minute or two.
All right. Thank you, Nav.
Just much happier that you're the one closer to the wall than I am.
Yeah. The water here is not very clear. You can
It's
see
not.
my lasers through the whole thing.
Yeah.
So Scott France from University of Louisiana Lafayette has just made a really interesting comment. Oh, that
Right,
he
I'm
may
moving a
actually
little
call
bit
in and
now, I
tell us
think.
about. Scott,
What was that?
I'm starting to move.
Yeah, that's good. This thing still doesn't show up.
if you
on
don't
your
want to,
sonar.
I can
Nope.
Guess you're
do
above
it
it.
for you.
Okay, I'm going to try to see if I can get close enough now.
Can you hear me?
Yes, we can.
Might be able to do it from this
Hi.
distance.
Sorry,
Keep the light in
the
the
video was broken up. So
place.
Okay.
what I was typing here in the
All right,
event
let's give
log
that a
was
try,
that we've done a number of ROV dives on seamounts in the North Atlantic, and some of which have some fairly steep walls associated with them. And very often we see on these walls, at least on the lower parts of the walls and the mid parts of the walls, that sponges seem to be the dominant biology, and in particular vase sponges and some mounding sponges that I really haven't seen very much of here in the Pacific. And then as you get to the higher reaches of the wall, not too far down below the top edge, you'll start to see corals become more abundant and larger corals, and they will replace some of these sponges. They'll intermix, but the corals become more dominant.
Would you want your
And then when you
sonars
get
in
right
good
up
well?
at the edge, right up the lip,
that'd be good.
the
Got
coral abundance and diversity increases, the density and diversity increases. And then behind it, if it flattens out, you start to see a different kind of coral. So perhaps some of the sea fans like plexors and so on. And so it'll be instructive and interesting to see if we see a pattern like this as we go up this sharp wall here on this feature. And I see we're looking at this beautiful vase sponge, and so I'll turn it over to Chris and Shirley.
Thank you very much, Scott, for that
I'm going to try to come
little
in
insight into
closer
what's
here.
seen in the
I
Atlantic.
think this sponge you're looking at now is a new genus in the subfamily Corbitellinae. So it's been collected, and it's a soft sponge, not a hard one. I was thinking it could be hard, but now I think it's soft. It's in the family Euplectellidae, and they're characterized by having this big, huge, giant osculum. That's the big hole there right in the top of it. So we're looking straight down inside of it. And on its sides, it's got all of these little projections with smaller osculum, and the water's probably coming into the side of the sponge. The water's probably running along the slope, and then it's exiting through this big hole that we're looking at right now. So these are always pretty interesting to me because there is some speculation that they're taking advantage of a Venturi effect in that water running across this big opening is actually pulling water through the sponge at a faster rate, sort of like when you're, I guess if you're blowing over the top of a Coke bottle or something.
All right. Go
Very
right.
cool. And so right next to the sponge was a Chrysogorgid from a Plurigorgia genus. So that was really interesting. Thank you, Chris.
I just wanted to add about that sponge. We've seen sponges kind of like that up in the Aleutians of Alaska, and I didn't hear Chris say this, but each of those little tube push-outs from the wall had an amphipod living inside of it, so we kind of described it as a little amphipod hotel.
We'll do that in a minute. So what? Do you think Sirius has stopped moving?
No.
No. Okay.
Yeah, Sirius might still have maybe.
So I
2 minutes
really appreciate
swing.
that close-up because I'm not sure I would have gone there if we hadn't gotten a better look at it. So that really helped potentially identify that animal.
Great. Always happy to help.
So it's less steep off to port.
It's strange because it looks like Sirius is picking up D2, but not the feature.
Yeah, it's really strange.
There's the feature.
Oh yeah, that is not D2.
Did you just come down a little?
Not right that second,
Oh.
but a moment or two.
Roland, were you saying something to me?
You coming up?
Oh, interesting.
A little bit. Yeah. I'm just kind of looking around.
Right.
Waiting for you.
Now, if we want to make the top and give you room to stretch out, we should come another 5 or 10 meters. If you want to be cautious about it.
There are many stalk-looking things on the left edge.
See those guys?
Yeah, so I think that's all sediment trapped in the sort of ledge, a sheltered ledge.
Pilot, do you want your mini Zeus aimed up to see if there's any overhang? Or are you
I think we're good.
okay?
Let's see here.
So here we can see a couple of sponges, likely Cornulus matinee, as well as some Tridopluras,
Coral on the left
probably,
edge of the
just coming
screen.
into view.
Interesting.
And again, you can just envision these pillows starting to sort of run down the sides of this ridge. This is totally not what I expected to see today.
All right, you're going to run out of tether soon that way.
Okay.
Yeah, and pilots, according to HiPAC, we almost have a 100-meter offset from
Sorry, Roland, what was that?
what we're actually seeing.
In depth?
Yeah.
That's unusual. Usually the depth is spot on. Where'd this
Okay.
Maybe the canyons are throwing it off too, though.
Yeah, I guess so.
If the HiPAC
What's
just
right
hit
in
that,
front of
maybe
us?
the top or
The
bottom.
coral?
Well,
And it's
well,
like
yeah,
a
think about
sponge and
I
a
mean,
coral
the-
and what's the other thing?
the
Another
sonar
sponge?
should only return the
Or is that part of the
shallowest
same
depth in
sponge?
the pixel, so
Little
Oh.
fish.
Yeah, that makes
Roland,
sense.
can we zoom in the
Video.
upper?
We going on this fish?
Oh, where's there a
Fish?
fish?
Video. Can you zoom on the fish?
Where is there a fish?
The one time.
Oh,
you
yeah, I
want-
see it.
I don't, as usual.
Yeah, there's a fish in the middle.
Eagle eyes, how'd you miss that?
Yeah.
So this looks like a type of,
Discussing
oh,
other
I don't know.
operations.
Need to
Coming
get
over.
some more zoom there.
Co-pilot, how do you
Does it
think
have
you're
a
still
dorsal
swinging,
fin?
or how do you
It
feel?
does,
I think
yeah. Okay, so I was
think
going
I'm
to guess
stopped here.
macroid, but I couldn't
Okay.
see the dorsal fin properly.
That
But
agrees.
if that's what Bruce is going with, I'm going to go with that too.
Coming left.
Still got a lot of zoom left.
Yeah.
So these macrourids are also known as rat tails or grenadiers.
Can you go a little wider? Find him again. Coming down.
And unfortunately, we didn't get enough of a close enough look to tell what genus that guy was from.
Maybe push in.
Yep, you can push it now.
I didn't even get that.
Okay. Yeah, I'm tugging around here. Can we go wide? Puppied off.
You're still in my view. I didn't
I'm
chase you.
amazed at the dramaticness of
Yeah, but I'm
this.
so far down the hill from
Yeah, the topography is much more
Altitude
interesting
lock
than we had expected it to
to-
be.
Incredible.
I mean, when you look at the
Okay, so
even
I'll
the
get off ahead
the
now.
higher res bathymetry that we collected today, which was 50 meter gridded, so that's not, it's not at this resolution level. We really didn't expect to see this.
What I like to do quite a lot when you're doing deep sea exploration is sort of
So it looks
imagine
like
how
you're
it
at
would
a
look
little
on land.
Nose.
Yeah, I'll come around. So
Yeah, this would
I'm
probably
in line
be
with you again.
a place of interest.
So I'm just
with
worried
these
that if we run straight ahead, we're going to
strange
run
pillow
along
lava
the nose and not
cliff
up the nose. So I think we
things.
should come a little more to
Do you
the
see
west
that anywhere on
than our
land? Probably
last
not.
move.
In
The only pillow
Copy, so
lavas that you
maybe-
see on land
Uh-huh.
are pillow lavas that were formed in the ocean
and then,
and then
Maybe we're gonna
abducted,
run right up
so
that
basically
mountain, yeah.
pushed upward
You could do
Right.
maybe
Um,
220.
during tectonic processes.
That might
Right.
send you on
So because
See what he
pillows can
lines up
only
with.
form in a
In the
submarine
ocean.
environment. Yeah.
interesting.
And often the areas where there are very good pillow outcrops on land, they're thought to
Quite a
be
nose.
analogs for
Yeah.
the deep sea.
Am I
And
up
so
to
we've
the
often
200?
used them as teaching
much.
tools to try
200
to
D2s,
understand
or Cirrus is
what
looking
is
maybe
happening in the deep
210
sea.
at you.
Yeah.
So do you want to do a 210 move of
Great, Dev. Thanks.
Yeah, I think we can do
And
like
Scott
30
says
meters.
that he would
30 meters
gladly
of-
hike in this
I think
terrain,
we're clear of everything.
I
yeah.
would too.
especially if
agree
it had
with that?
land corals
Yeah.
and
Let's
see
associates,
30
i.e.
meters at
trees
210.
and birds.
Thank you, Nav.
coming up.
And while I wait, I'll just sidle down a little.
Bridge,
and look
this
at
is
these
Nav.
corals here.
Sounds like a plan.
Hi, can I get a ship move of 30 meters at 210 degrees at
Those
0.2
sponges lower
knots?
center, there's a rock or something with vertical lines on it.
Good copy. Thank you, Bridge.
I wonder if we're starting to see the sort of the base of this broader ridge that we were trying to reach. It's very hard to tell based on what we've seen so far in terms of the bathymetry.
Yeah, I cannot tell you what we're seeing for bathymetry.
Hi guys, it's Scott again, and I see we're more or less at the top of this particular feature, and it's possible the edge wasn't sharp enough to get the
Hey,
really
Bridges, Snap.
concentrated corals on the side, but I can certainly see lots of corals on the top.
Yeah. So this sort of goes
copy that. You
with
can
what
go
you were
ahead.
saying about the zonation, where you would see the sponges further
do you wanna
down and the
zoom on
corals
these
further up,
spires?
right?
Just an FYI, ship's rotating starboard 10 degrees about A-frame.
That is exactly correct. It just there
Thank
was
you,
no
Dan.
band that was kind of around the edge, but that might be because of this smooth transition. So the reason you tend to see lots of corals and sessile fauna on those edges is because the sharp edge accelerates the current as it sort of moves over it, just like an airplane wing. If the airplane wing is curved, it forces the air over it to move faster above than below. And so that's the same sort of process that's going on here.
Okay,
And if the
go
water
ahead,
is
yeah.
moving more quickly, it means more food per unit time is being delivered to the coral.
Yep. And in this case, what we saw was really rounded edges, probably, mostly due to that manganese crust. Although some of it might also just been drooping pillows coming down the sides.
I'm just going to peek down here.
By the way, I'll pass on that suddenly the telecon is much clearer. That's nice.
Oh, great.
That's good, yeah.
Wow.
So is this sort of a local high we're looking at here?
Yeah, we came to the top of this kind of nose.
And are you still surrounded by walls?
No, we don't see any more walls.
Okay.
The ship is moving 30 meters this way now. We're going to start moving again.
Great. Thank you.
You want me to hold heading or follow you over?
I guess follow me over.
Okay. I think we're starting to tug each other.
Yeah.
I can come down a couple meters if you want.
No, you're already pretty low.
Okay.
I'll come back to center.
Sorry, Nav, are we doing 210 or 200?
We were doing 210.
Thank you. All right, I'm going to come back on the heading.
Okay. Now that I'm a little closer, I can actually go down a bit to look.
So, Shore-Based Science Team, is there
Yeah,
anything
also
right
getting
here
the
that you'd
tilt
like us
down.
to zoom in on?
There you go.
I've got a tilt down, or Mini Zeus is looking straight down the wall.
Yeah. it's all
Ooh,
boxed
look at the
up.
view in
Are
the
they
Sirius.
just waiting for the ship to catch up or something? It seems like we're being here for quite a while.
I believe that's true, that we are waiting for the ship to start moving. Is that right, pilot?
Yes,
Yeah.
the ship has been moving for a few minutes, and Sirius is on its way.
Yeah, so Chris will be starting to move
Yeah,
in a few
co-pilot,
minutes.
you should start to feel swing soon, if not yet.
Great, thank you.
Yeah, as the vehicle, as Sirius has been getting closer, the pilot has just been going around the edges of this nose projection that's coming out to give you guys views of everything that seems to be surrounding it.
Deb, as we are sort of moving over the substrate here, coming up on the upper left of the screen, I see there's some shortish fan-like or colony-like material. Maybe we could have a close look at that.
I also now have sonar return.
We can certainly try.
20 meters or so.
Are there?
Thank you.
Well, I'm glad you do. Means your sonar's working again.
So, pilot, can we
Yes.
look to the left? Did you hear that?
I heard some fan-like.
Yeah, that's
Is it
what I heard too.
These brownish ones, kind of on the edge.
I'll try to give you some guidance once the video image clears up on my screen.
Great. I think that would be
Suspect
helpful.
It's the ones coming left of lasers right now.
Yeah, that's what I was headed
Yeah,
for.
I know my description wasn't very clear.
The ones.
So I think he means the ones on this rock right in front of us,
Okay.
perhaps?
Okay, video. Free to zoom.
And then possibly ones
Yes.
down the
Yes,
left.
Scott,
Eva,
is
I
it these?
believe you are correct.
Yeah, so
Oh,
these
good.
Antipitherians, these are black corals.
And remind me why these are called black corals? Is it because the
I think it's
skeleton
because
is black?
The skeleton is black,
Okay.
exactly. And so they used to be used quite a lot in the jewelry trade.
They are also known as thorny corals because the skeleton is also very spiny. And I guess I can also throw in the name Antipatheria translates to against disease. And back in the days of the ancient Greeks, they would, and still probably, they would dry and grind up the skeleton and use the powder for various medical purposes.
Wow, that's a great fact.
That's really interesting. Does it work?
No, I'm going to assume it's like...
Well, I mean, because now they
Rhino
have all these,
horns
but
and-
now they have all these drug discoveries that come from sponges. So I was just wondering, like,
So Tina said that this is likely trissopathies, the Antipitherian trissopathies.
Just another minute here.
And you can see
Okay.
the very clear black
Nice,
skeleton.
All right. You can go what?
Scott, did you want to look at any others of the black corals?
Sorry, Diva, sorry for the delay. I have to move back and forth between the phones. No, if all that little patch is black corals, that's fine. We can move on as you would like.
Thank you.
Thanks for the
Yeah,
close-up.
co-pilots, you should start coming up to keep our delta higher.
Coming up high.
Can we go with a partial zoom as you go across the surface?
Sure.
When you're ready.
I'll try to stay lower to get the light on. Okay, video, you can zoom.
Maybe just a little tilt down. Just keep the top in the air in the scene, but a little closer view of the coral.
So again, some more of these black corals. They're the sort of yellowy branching ones. There's a stalked crinoid also here, as well as some types of octocorals with comatulid crinoids on them. It's amazing how different this community is from the community we were seeing yesterday.
But we've definitely entered a region where there's a lot more.
A lot of black corals, that's for sure.
And we didn't see a ton of black corals
No.
yesterday, did we?
No, not a lot at all.
Unfortunately, I'm not seeing anything past this nose. Might have to go off into space again.
I think you guys just made an observation that we can make a point on, and that was it's interesting how different some of what we're seeing today is from yesterday, and indicates why it's so hard to characterize some of these areas with just a single dive. We're actually only getting to see a very small snapshot of some of these deep-sea seamounts and ridges and volcanoes and so on.
Can go in
And
closer
as you can see, there's just so much to see, and every day we're excited about something that we've seen that we didn't expect to see, or something new, and always a joy to be down here.
Absolutely true,
Well
Scott.
said. Well said, Scott.
So this is another dead sponge, I assume?
Exactly. So this is a rather large dead sponge, and as you
Spot
can see,
lobster?
you've got hydroids growing on it, a squat lobster.
We're still getting close.
Yeah.
That's made
Coming
its
back up
home.
for you.
Yep, pilot. So on high pack, it's showing us should be 2250 about, and Sirius is saying 2150. So there is like a 100-meter offset in depth if this is the shallowest point we're
We
at.
should double check that the contours are.
Yeah, or the contours were just mislabeled.
I'm also
Pilot,
showing
when
a
you're done
60
zooming
meter
here, can we
altitude
zoom
right
down
now,
near the
so
base of this?
Sure. Can you go wide
Actually,
video?
for the other things around it. You want to zoom in on the little sponges?
So I think in this terrain, we're honestly within the margin of
Just
the
for a
error of the
moment.
sonar.
Yeah, if we get if we're waiting,
Yeah,
then
about 100. Or even if the well, I guess the
I think
offset
they're busy.
equates to the. But 100 meters in depth is usually indicative of something else.
Well, are we actually on the 50 meter grid that was made this morning, or are we on the 100 meter grid that we had previously?
I think we're on last night's.
I'm not 100% sure.
Yeah.
All right.
Okay, video. You want to zoom on these little sponges?
Steve, are those
Going for the
the
white.
same sponges?
exactly.
Or,
What I
oh,
was
they
just
have the
about to
little
say.
they have the little
So-
white spots too.
So you can see that
Yeah,
these are the
Copilot.
same
So
Down?
sponges that we
you
just
might have 2
collected,
minutes
as
left
well
of
as there are
swing if
some
it's similar to the last move.
tiny white
Just heads
stylosterid
up.
corals, I think, in between. But these sponges are just
Look,
like what
shrimp.
we just collected. Oh, and a nematocarcinus shrimp. But
I don't know if you're right.
why we collected these sponges is that they have these weird white specks, which you can see inside of them, which we all found quite confusing. We weren't really sure what they were. But you can also see this nematocarcinus shrimp, and they have the really characteristic long limbs and long antennae. And then you can also see some Ophiocanthidae brittle stars moving around in the background.
Some focus. Okay. Can you go wide?
Yeah, just following the science.
Yeah.
I'd love to get a much better view of those white spots on the sponges.
We did collect one.
Yeah, we collected one.
Yeah, but I don't think we've actually imaged it really well.
Yeah, thank you, pilots.
Welcome. Okay. Can go in and get a close-up of that.
So this community is sort of dominated by sponges and corals, but not in the way we expected. A lot of the
It's max zoom.
seamount areas off the Hawaiian Islands and the ones we'd visited before had quite large sponges and quite large coral colonies, like of isidids and primnoids and other types of large branching corals. But here we've got this very different community which has smaller antipotherians or black corals, smaller sponges,
I'm not sure if I can answer their questions.
and not that many of the large branching octocorals.
Okay, whenever
It's
you're
interesting.
happy video.
Hi.
Let's figure out where the next land is.
Well, I got returns all to the south.
That kind of agrees with our terrain.
Yeah, that's somewhat correct. That's pretty good considering, I guess.
I've got nothing. So what do you have?
It looks like it's all to about.
Do you want to
What?
turn
Pilot, can I put your center
turn
out?
that way?
I
Yeah.
put it in earlier.
Oh yeah, please put mine out. I didn't know.
You're only seeing 30 meters right now. I'll put it to 50.
Tilt down, pilot, as you're looking around.
Yeah, even at 50, you're not seeing anything. Do you want me to put it out to 80?
Yeah, let's try.
Interesting.
So you swinging your heading around?
I am coming
All
out
right.
due south.
Thank you.
Yeah, at 80, you're still not picking up anything. So you might be at
Okay.
just a really local high.
Yeah. Well, luckily.
And then Sirius picks up some stuff.
How
But is
far?
that
south?
Oh, that's probably. Yeah, that's me.
Oh.
25 meters out. Yeah. So yeah, we have nothing on either sonar. Well, watch leads. We've come to the top.
Yeah, we're listening in on you. So basically, you're at. Tell me if I'm wrong. You're at a local high that we can't really correlate particularly well to the bathymetry. Is that
Exactly,
correct?
yeah.
Okay.
Yes, this very steep nose.
Yeah.
And, we have both of our sonars
And you see
set
nothing,
out
right?
to 80 meters,
Yeah.
and there's nothing. So, I guess what we'll have to do is just trust HiPAC that there's more uphill to the southwest and just kind of move that way.
Are you going to wiggle around this or over it?
Well, we're already on.
Like
already
you're at
on
the top,
so
so just going to
yeah. If there
go
was
over
more
it?
up, we would go. Yeah, we're already over it. Yeah.
Okay. All right. I will relay that to the shore-based science team.
And
Maybe-
off into the abyss.
Should we set Sirius sonar out a couple just to make sure? It doesn't
Yeah.
usually pick up much, but
Do you want to put yours in or leave it at 80? So Sirius is at 80.
try moving it to 100.
So shore-based science team, I'm not sure if
both of
you've
them,
been
just
listening in on
See
the
if
ROV
different
feed as
frequencies
well.
give us anything.
We're in a kind of an interesting situation in that we had been moving up these very steep walls. We found ourselves at a local high, and right now, in terms of the sonar, we see nothing around us. So
Yeah.
we're at a local high that was really unexpected for what we're seeing on
That-
the
It's
high
It's-
pack
it-
bathymetry.
it's 3 sweeps have shown
So
that same something. So
what-
maybe
it's about the right
4
direction.
sweeps.
So they're
Yeah.
probably going to have to go
That's-
What is
over
that,
this
about
little
200,
ridge
205?
and try to see if we can
Oh,
acquire
let's see,
sort of the
one-
main bridge
yeah,
that we had been hoping
so,
to find.
Yeah,
Do you want to do a move towards 205?
Yeah,
yeah.
but that's 60 meters away.
Yeah, why
Yeah,
don't we do
let's
that?
do,
Let's just keep
let's do
moving
a 40
forward
meter.
and see what we can see.
Yep.
And yeah, that might be it.
40 meters at 205?
Yes, please.
Copy that.
That agrees with.
Yeah,
The
that
high pack.
makes sense.
Yeah. All right.
Bridge, this is Nav. Hi, can I get a ship move of 40 meters at 205 degrees at 0.2 knots? Good copy. Thanks, Bridge.
Carl, is your intention to go out into the blue, get Sirius over this and drop down, or are you plan to go straight to the next sonar target?
As Sirius moves, I'll take a couple minutes and turn around. But then after that,
Thanks,
I
Bridge.
think we'll just move ahead through the blue. Because we can't go, you see us, seeing how steep and jagged the terrain is, we can't really drop down backwards, or we'll never know what we drop down into.
Yeah.
Yeah, pilots, we have 30 minutes of bottom time left, so
Okay.
hopefully we find something.
Yeah. So the ship is moving now, correct? I don't-
Yes, ship
I
is
don't-
in motion, 205, 40 meters.
don't quite have enough tether to turn and look backwards yet.
Maybe take note of your altitude as you're going across. Sort of get an idea of the contours down below.
Yeah, I just put a point at that local high.
Okay.
And that had a, let's see, depth of 2180. And, yeah. And then in the I took one when you were in the canyon. That was 2180. It's strange.
So I think this is sort of both the joy and the pain of exploring sometimes. You come across things you didn't expect to see, and they're fantastic.
Oh, no, that makes sense.
But it sort of throws a little bit of a monkey
canyon.
wrench into
And then you came
the
up
plan you had
40
made
meters to
of
the,
what you expected
peak.
to see.
But I will add that this is the first dive we've had of this expedition out of 15 dives so far, where we've ended up somewhere where we weren't, where the bathymetry's totally thrown us off, the topography has totally thrown us off.
I could agree with that.
Yeah.
I mean, I think we've had not a lot of issues. We've had some issues with offsets between the bathymetry that we were given and what the ship and ROV were seeing, but those are pretty easy to correct for. You can kind of say, oh, I'm 50 meters off in this direction.
Think that's kind of expected as
yes,
well.
Yes, absolutely expected of when you're exploring in the deep sea. Spend a lot of time kind of dead reckoning towards points. Like, oh, look over there, and then figure out where we are.
So wait, we're going 210, is that right?
205.
205, okay. Co-pilot, can you...
Yep.
Oh wait, you might lose me then, huh?
I would have, yeah, probably.
Okay. All right. Okay, I'll saddle to starboard.
Copy that.
Get
I'll
centered
come over
up.
to 205.
So for those of you who are just joining us, and for our shore-based science team, you may notice it looks like we're sort of flying around through blue water. We are, but it's only for just a little bit of time. We were on a local high that actually we completely could not anticipate from our bathymetry. And what we're trying to do now is just move
05.
down a little bit and
Okay.
try to get back
Something up.
towards that ridge that we think we should
So,
be seeing fairly
when I'm in
soon.
blue water, if you keep your camera just about right at 45.
Yep, just got there. You're at
All right.
45 now.
Thank you.
I always feel like there should be
Probably
hold
heard all
music
this as
playing.
math before.
Yeah,
When we're
co-pilot,
going
you should
through the
also
blue
be
water.
starting to feel swing
Hold,
soon.
please.
I believe I am.
It is. It's fantastic. You're not on.
Why? This is all exploration. You see incredible life in the blue water sometimes.
That is absolutely true.
So yeah, 50 meters ahead now. Is that sonar return?
You're painting something now, too. Yep.
Yeah. Turn back.
I'm seeing it in the camera view slightly.
You see a little bottom?
I
That's
see a
good.
little diagonal line from the center of the camera up to the upper right.
Nice.
See more rocks in that direction.
Tell you what I didn't realize from not sitting in the seat is how much Sirius's heading wanders.
Yeah, if we're not a good distance apart,
Yeah.
it's particularly bad on days with current or big heaves.
Yeah, which we got a fair amount of heave today.
Yeah, not a lot of current, luckily, but
Can you guys yet see it in your monitors?
no.
No.
Okay. Seems like you've got another sort of separation, a couple rocks and some sand going in between.
Oh yeah, actually I do see something, I think. We're 21 meters up off the bottom.
I'm 40.
Kind of agree. When I was on the other side, it was 23, so that was just some sort of like
Pinnacle feature.
23-meter tall pinnacle that was
Yep.
in our way. We
Yep.
went up and over it.
I,
Yeah.
I honestly didn't even run over the top. I didn't show up with a shallow any
Yeah.
altitude.
Yeah, you were off to the side. That's why I couldn't really back down when we started on this heading. 'Cause, okay, I've got a pretty good return, 50 meters. It's a little bit to port now, but it's firming up.
And I think I saw the closest stuff more to your starboard.
Is the ship still going?
It's just finishing up. If
Another-
it just, it might have just finished.
Another interesting thing about today is the terrible visibility. You
Yeah.
can tell by my lasers.
Yeah, it's definitely cloudy.
I wonder if there's some activity around, hydrothermal activity, like even within a few miles it's blowing. Usually this deep, the water's crystal clear.
I would agree with that.
Yeah.
How long was that move now?
40 meters.
Okay.
Yeah.
So it's just yeah, it's about 40 now.
So shore-based science
Now
team,
we can see the thing
I
behind
think
you.
Yep. Do you want to
hopefully
come down
the audio is good
make
enough that
bottom
we can
contact?
attempt
Not
a
while you're
science
swinging, 'cause
call
then-
after we come
Okay.
off the bottom.
I mean, I'm above it
So
now.
we'll do as
It
we've
looks
been doing
like it
Pilots, do you want me to
for
bring
the
in
rest of
sonar
these
at all,
dives,
now that
where
we have some
we will wait about
targets?
Yeah, you
5
can-
minutes
yeah,
after we
sure,
come
they're
off
both at
bottom,
100.
and then we will reconvene and talk about the
Yeah,
next
that's
dive.
good.
Does that work for everyone?
Yes, Deb, you are sounding pretty good these last 10, 15 minutes or so, and not breaking up very much. Here is
So
why.
we're cruising at speeds like this. What's your average
Great.
forward thrust?
So we'll hope that it remains that way and that we can all hear each
I'm only
other
thrusting
on
about,
the science call.
15%.
So remember, about 5 minutes after we come
Is
off
that
bottom,
fairly steady,
I
or
believe we're
going
coming off
15
bottom in
0?
about 20 minutes. Hopefully we will have
It's
a
pretty
little bit
steady.
more time for exploration before that.
So the ship stopped, and Sirius is probably still swinging.
I think we're gonna need some more though, to
Yeah.
get us all the way over there.
Maybe I'll start. If Sirius is slowing, I'll start dropping down a bit,
Okay.
and you can follow me.
Copy that.
But
Follow you down.
we'll be ready to push ahead.
Sounds like a plan.
Okay, we can see some more terrain looming into view.
It's just like we were starting the dive all over
know.
again. Look, the bottom's in sight.
Except that we only have less than 20 minutes on down here.
I understand. So for those of you who are following along at home, we got a bit of a delayed start today. There was an issue with one of the fiber optic cables on the ROV. And so I think we lost
Still
about an hour and a
15
half,
meters
2
up,
hours
actually.
this morning.
Yeah, we still had a
Very
full
big
3 and a bit
fuzzy
hours on the
corals
seafloor
at the
there.
top.
And we all
Looks like
firmly
another
believe that
local
it's
high.
much more
Yeah.
important to make sure that we fix those issues and send the ROV down in a safe manner so it can return in a safe manner, even if it means we have to lose an hour or so of our dives. I don't see one piece of biology except whatever went swimming by just now.
There's some corals and sponges at the top. of
Looks
this
can be
offspring.
deceiving, Deb.
It's true. Our video engineer already told me there's actually corals and sponges at the top.
So as soon as you think Sirius is about stopped or close, we can move her in closer. I think another 20 meters or so would be safe. Do
Yeah.
you agree?
Yep.
Yeah.
I think we're pretty close to stop. You're
Okay.
moving out in my frame, and I haven't touched my camera.
Okay.
So I'd say let's start it. Because as soon as we start back up this, you're going to be out of tether.
So pilots, did you say you want another move or
Yeah, 20 meters.
20 meters?
Yeah.
Are you good with same direction 205 or do you want?
Yeah, seriously, still that way.
All right, copy. Bridge, this is Nav. Hi, can I get a ship move of 20 meters at 205 degrees at 0.2 knots?
You can see the
So we can see a Kamachlid crinoid.
Good copy. Thanks, Bridge.
As well as a sponge, which I can't quite tell if it's a Hyalinamatidae or Caulophacus, like a Hyalinema or Caulophacus sponge. But I'm sure as we get closer, it will become obvious.
Thanks, Bridge.
you can see the base of the wall in Sirius view.
Okay, video, would you like to zoom?
So that stalk to me looks like a Caulophacus rather than a Hyalinema. Chris, do you agree?
Pan down a little bit.
No, I don't think it's a Caulophacus, and I don't think it's a Bolosoma either. But it might be more Bolosoma-like. It's hard to say where it's attaching. Anyway, this is kind of a strange one. It's pretty interesting.
Good. We like to bring the strange
Alright.
sponges to you, Chris and Shirley.
Keep moving since we're short
And
on
I'll
time.
note that, again, we're looking at that fairly heavy manganese crust with that very pebbly texture.
Pulling for
Light
wide.
sediment, though.
Thank you, video.
A little more moderate in the pockets.
Did we look at this coral looking thing? That's this thick. It's got like slight split at the top on the right of the lasers.
I haven't seen one of those today.
Seen it in previous, but not in a tight look at the top of it just to see if it's coral, crinoid, whatever.
Now, as we're creeping up the side of this rock wall almost, we can see
What is that right in front
this
of
looks
us?
like it used to be a crinoid, but the crinoid's arms have all broken off.
Okay,
That's very
video.
sad.
Can you zoom?
Yeah, see, look, they're now regrowing.
So it's still
Cool.
alive.
Coming
I think
back up.
so, yeah, and it's got a healthy colony of hydroids on its stalk. But if we were to look back at the calyx and arms, yeah, all of its, I'm not sure what happened to it, but it's slowly regenerating all of its
Who-
8, 9 arms.
Who eats
10
crinoids?
arms.
I mean, is there anything that predates on
Fish?
them?
I don't know.
No.
Anybody on shore know?
I'll watch the event log for an answer.
Okay, pulling out.
Okay.
Fish, evidently, is the answer.
Yeah.
So let's see. With this heading, you're not quite lined up with me in the local high.
Correct.
Do you want to? Let's see. The ship is stopped, correct? Yeah. So maybe.
I think it's just finishing up, actually.
Maybe turn
Oh, and
this
as
way.
we're
Maybe,
moving
yeah,
up, we
it's
see
just about
we're
to stop.
I don't think
sort
you'll
of in
slide much
this
more
sort of
sideways.
coral garden
Yeah,
area.
anyway,
How
so
nice.
I'll come over to face you.
So this is much more like what we were seeing yesterday, but have not yet seen today until now. Though there are still quite a couple Antipatherians in between. Okay, so if we can get a couple views of these corals, that would be great, pilots.
Sure.
So the Antipatherians we've been seeing are Trisopathes. These are yellowy, stumpy sort of corals. And then all of the others are Isidids.
So let's see what's on these are maybe an enemy or two. Couple of voids.
Okay, video, you can zoom.
Thanks, Bridge.
Okay, so this looks like an Isidid. We can see it's very defined branching, okay? And it did have some commensal actinarians or anemones on it.
Tilt up.
Pilots, we have about 10 minutes left. Would you want to get a last ship move in, or?
I think there's a lot of coral around
So
here.
onshore scientists, I'm not sure if you heard that, but we have about 10 minutes left in this dive.
Okay, what else do
Is
we
there
got?
anything in this area that you would like to get a good
Definitely
look
not
at?
Facing us, but I don't think any are.
Yeah.
I would think a short move just to give you a little
This
bit of
one?
tether to focus on things would be worthwhile. I don't think you can go very far off that one spot.
Yeah.
Would you want maybe a 5-10 meter move forward?
I'd say 10-2-3-0. Carl, you
10,
okay with
2,
that?
3,
I'm going to
0?
take that
Sure.
to be a no.
Copy.
Okay,
Um,
just look
oh.
at a brittle star real quick. Either of the other two.
Bridge, this is Nav.
And this coral's also got some Ophiocanthid and Ophiuroids living on
Hi,
it.
can I get a 10 meter move at 230 degrees at 0.2 knots, please?
So pilots,
copy.
we've
Thanks,
just heard
Bridge.
from our onshore team that for the last 10 minutes, they just like us to continue getting really good close-ups of different animals.
Okay.
So I think behind this coral, there was another type of coral in the background, which, ooh, is he going to leap? Is he going to go for it? Oh,
Just
oh,
walking
oh.
around.
You can do it, Guy. Come on.
Don't do it.
It's
Hi,
like
guys.
watching an orangutan.
I had got disconnected from the telecon, so I was calling back in when you had that close-up on the bamboo, and now I'm sitting back here watching. I see that Chris was giving me a hard time, but I'm not sure
Okay.
what the question
going
was.
full volume.
Trying to identify this.
Maybe it was,
Black
yeah, to
one.
try and identify this Isidid we were looking at.
Left, if they're interested.
Okay. Watch
Yeah,
out.
that was the question.
Is it this
What a
black coral?
surprise. I suspect this is in the clade of that we're calling the J clade because it has this newly described genus of bamboo coral from
Okay.
2012 called Jasonisis, named after the ROV Jason. And that was discovered down off Australia and around New Zealand. And there's actually a lot of diversity of bamboo corals that are related to that genetically,
Just go ahead
but
and
we
tilt
don't know
up.
what they are. We haven't described them yet. It's a
Hard
big
stuff
project
to
ahead of
know.
us, and there's quite a range of morphology, so they're kind of difficult to identify in images, but I'm basing what I just said on some collections we made in Hawaii last year and some sequencing I did of that material. You can see that it branched at the nodes.
Okay, we'll pull out
Something
and
about
we
the
can find something else.
appearance of the tissue when I look at it and the sclerite. So that's a guess. It's either that or in the B clade. And when I say B clade and J clade, what I'm referring to is we've done, one of my graduate students, Spree Heestand, who's defending her PhD next week, has done an analysis of several hundred bamboo corals based on their DNA sequences, and you can arrange all those sequences into a tree form so that you see which ones are more closely related to the other. And groups of those that have a common ancestor and a morphology that
Is that a crack
we
in
can
Sirius's
identify, we
view
call
on the
clades.
left?
And so instead of giving you the
It's
genus
just a
name
small
like
ridge.
Keratoisis or Lepidisis, we refer to it as this genetic name as a temporary placeholder until such time as we can get to identify it.
But
So are you going to be able to
I'll look
name
over
all
there
of those
to
clades?
survey these corals.
Scott?
Somebody will
Crack,
get to
actually.
name all of those clades, and right now Les Watling and his student Abby Lapointe are hard at work on doing that for a number of them. Abby's got a number of names just waiting to
crinoid hanging off of that one.
be published. And so, yeah, there are some interesting creative names that we're not supposed to reveal until such time as they're published. They've gone through a peer review.
Okay.
That's the typical standard, is you don't
just
use the
peer
names
over, keep the
until
delta
they're
like 18 to
have been
20.
reviewed by other taxonomists, formally published, and then you can use the names.
Great, thank you, Scott. And this is why, you know, expeditions like this are one of the reasons why they're so important, in that we only collect two biology samples or so per dive, but they can be extremely valuable in helping to determine, helping to define these species groups, basically. So this is another camatulid crinoid from the genus Sarametra, and you can see here it's using its many cirri to hold on to that coral branch. And where it's holding on, it's actually missing polyps. So perhaps this crinoid's actually sort of damaged this coral in that area, and you can just see the tissue's been worn away, and now it's just a skeleton
Pulling
left.
for full specimen.
Okay.
Took down.
10 minutes. And there was also a little anemone on the coral there.
Is that a group of anemones down in that direction? Pan to the right a little bit.
And like
I'm looking
Pigafetta...
at the screen. Sorry, go ahead.
No, go ahead.
I was just going to say, as I'm looking at the screen at this this beautiful image, and I'm studying the coral, I also have my computer open here to a database that has the bamboo corals that we collected last year, and it also has some of the microscope imagery of the collections that we made. So when we got them up on the ship, as you guys are doing, we put them under a microscope, take lots of photos, so we have very good coverage of these things. And this
It's
looks
just a
fairly
couple more
similar,
minutes,
not
and then you should
identical,
start
but fairly
setting up.
similar to one from the B clay that we collected at 2,255 meters depth
co-pilot?
on the mid-Cairn Ridge.
Make sure.
And we are at bang right about that same depth level. So it's fascinating. A lot of these species only live within certain depth intervals.
The starboard lower goes all the way right first and all the way
temperature
starboard.
and something about the water masses themselves.
All right. Starboard low.
So first you would find it with this camera. If you hit one enter, you can find it.
You'll rise up a little bit, pilot. Just rotate around as you guys prepare. Tilt down.
Just going to take a look at the backside here.
So all the way, sorry, you said all the way right or all the way left?
They're spread out all the way as their home position. So starboard goes
So
all
in
the
our
way
last
right.
few minutes here on the seafloor, I'll note that like Pigafetta Guyot, Enrique Guyot is also named after Magellan's voyage. Enrique of Malacca was Magellan's servant and interpreter who was able to communicate with the different tribes that they encountered.
Huh.
Yep. And while it doesn't say it, I think it was actually proposed by the same person who named Pigafetta Guyot.
Wow, okay.
They were both, the names were accepted and accredited at the same time.
Starboard side looks
That makes
good.
sense.
Yeah. I think we're going to learn more about Delcano being probably
Tomorrow.
possibly part of this as well. But maybe not.
But
we
you
don't know
stay
yet.
tuned because we're not going to reveal that till tomorrow.
Yeah. Interesting place names on the seafloor.
So looking out here, a couple more stalk sponges, some icidid corals, some black corals. What looks like at the very top of the picture, a stalk with a camatulid crinoid on it. There's quite a large dead sponge just going off the bottom right corner.
I mean, I know we haven't seen many squat lobsters, but I sort of feel like this would be the place to find it, playing King of the Mountain. Do you know what I mean? It's not here, but
yeah,
it's where we always find them, sitting at the top, like, hi, I'm a squat lobster.
Definitely.
Swing arms.
Not in this
But
case.
yeah, there's been, the ones we've seen on this dive have only been commensals.
Mm-hmm. True.
Hydraulic secured.
Okay, Nav, is it time?
You can look around a few more minutes if you'd like, but
Camera down a little bit. Off
yeah,
to the
just a
right.
couple minutes.
Can we zoom in on that coral that's just gone off the screen below? I think it was a Bathypathes.
You want to follow me around to Copilot?
Bathypathes.
Science
Actually, let's get a
outdoor
zoom on these
From-
ophiuroids before
Pilot,
we. Sorry, dead ahead on the stalk,
Science, I
right
think you
ahead
need
of
to
us,
key
just
up
because
the
it'll
pilot.
probably be pretty.
No, I
And to see what's
can
at the
hear
top
you.
of the stalk. Is that actually a sponge?
So which
Ball?
which coral?
The one directly dead in front on the top of the mound
Oh,
with the
the
crinoid
yeah.
that's now opening up.
So, pilot, I seem to have lost control of my rotator.
You're up and down.
Yep, on Zeus.
It's just very touchy, probably.
If you pull too fast, it won't respond.
All right, there it goes.
Yeah.
Okay, so if I pull too fast, it was on. Good to know.
Yeah.
All right, following you around,
Thank you.
Yeah, pilots, it almost looks like another valley right
Yeah.
after this.
I see that. It's a good thing we don't have to do another jump.
So this is another Sarometra crinoid.
Okay, but here you want to zoom on the top.
And on
here?
top of this, the stalk, it looks like there might be a
See
tiny.
there, just 1 minute, pilot.
Wow.
Okay.
So is this
Sorry.
current because of us, or is this what?
No, this is when we came up over the top
Ah.
here. There's a kind of a new current
Interesting.
that's bouncing us around.
And as well as you can see, this there's a,
Gonna back
a
up a little
sponge.
for you, Vinny.
It's on a sponge stalk with a very very
Come down to these.
small
Just adjusting focus, not zoom.
Okay.
sponge appendage bit, and then there's also some Ophiocanthidid ophiuroids,
coming back up.
very very tightly wound, around their sponge stalk, which probably indicates how strong the current is here.
I wish I could wind myself around a sponge here. Okay.
They seem
Yeah.
very flexible.
Great, that's awesome, pilots. And then just to the right, sorry, one last thing before we
Of
leave.
course.
Just to the right, below this sponge stalk, there was a Bathypathes coral, and they look a bit like a palm tree leaf. So
Gotcha.
a central stem, and then leaves fringing
out.
off either side of
Alright.
it.
Like a stalked crinoid, but not.
What? No. What?
Oh, I think a stalked crinoid looks like a palm
Right,
tree,
so it's just there, just above the bottom of the screen.
Think just one arm of the stalked crinoid.
So one leaf
One leaf of a palm, yes.
down in the bottom or left, bottom left there.
Yep, come into center.
Yeah, pilots, this will be the last zoom, unfortunately.
Okay.
Yeah, so it's on sort of a longish stalk, kind of. Just your lasers are exactly on it
Yeah.
right now.
Okay. Want to zoom their video?
Can we get a zoom? Yeah, that'd be great. Thank you.
Coming back.
So, Tina, if you're watching right now, we'd love to know what kind of Bathypathes this is. Perhaps Patchoula? Just beautiful.
Next zoom.
Okay, that's great, pilots. Thank you. So we are
Alright.
done. Whenever you are
Okay,
ready to
video.
come off the seafloor, we
Yeah,
are happy
clear.
to come off the seafloor.
Okay, we're ready. And you can see we came to the edge of this hill, and if we were to continue, we would look around and then jump off again into another abyss.
That's interesting.
Yeah, very interesting.
It's off to your right.
Yeah, and and watch leads. Where we're at right now, we should only be seeing a 10-degree slope.
Oh.
So I think high pack's a little off.
I think we can all agree that the high pack and what we saw today were not
This is
the
just
same
amazing.
thing.
Look at that over there.
Yeah,
Yeah.
not the same thing.
I'm going to have to agree with Scott. This would be an awesome place to have a hike.
I would agree. All right, thank you guys.
Yes,
Thank
thank
you,
you.
RV pilots. Great job today, as usual.
It was our pleasure. Thank you for putting up with all the weights. Blue water.
Yeah, we didn't want you to run your vehicle into anything.
Thank you.
All right, so shore-based
All
science
right.
team,
I
you've got
basically
about
just need to pull
5 to
180
7 minutes
port,
before we
and
get back
you
on
just
and
got
chat
out. I
about
think we're in
tomorrow's
great shape.
dive. So
Okay, so the
please
tether's
come back
under you
soon.
for your turn.
Thank you.
I believe so.
Yeah, you're 20 up. Okay.
You want to? Can we go ahead and do that now, or a little bit and do it, or you want to
I'll just
stretch
come
out?
up.
Okay.
I'm just
Video,
gonna come
this
up
is
like,
Watch Sleep.
5 meters or something,
Sounds
just
good.
so I'm a little safer.
I'm going
But
to come up a couple meters too.
I'm
okay.
doing kind of awesome over here, actually. Could you please disconnect our party line from the stream?
Okay, we are
Thank
off
you.
bottom.